In this video, you will learn about the structural and functional properties of neurotransmitters. Neurotransmitters are fast-acting chemical messages released when an action potential arrives at the axon terminals of a presynaptic neuron. Neurotransmitters diffuse across the synaptic cleft and bind to specific receptor proteins to produce rapid effects in the postsynaptic cell. Typically, the receptors will open ion channels in the postsynaptic neuron, producing graded potentials. Summation of graded potentials allows the postsynaptic neuron to integrate information from multiple inputs. If the postsynaptic cell is depolarized enough to reach the critical threshold value of negative 55 millivolts, an action potential is triggered in the postsynaptic cell. Receptors for neurotransmitters come in two main forms. Ionotropic receptors are ligand-gated ion channels that directly cause graded potentials. For excitatory neurotransmitters, these channels allow sodium or calcium ions to rush into the postsynaptic neuron, producing a burst of depolarization known as an excitatory postsynaptic potential. For inhibitory neurotransmitters, these channels allow chloride ions to rush into the postsynaptic neuron, producing a burst of hyperpolarization known as an inhibitory postsynaptic potential. Metabotropic receptors are G protein coupled receptors. The G proteins activated by a receptor stimulate production of second messengers in the cytoplasm of the postsynaptic cell. Second messengers could regulate enzymes or ion channels to produce slower, longer-lasting effects compared to the graded potentials produced directly by ionotropic receptors. Now let's look at some specific examples of neurotransmitters. Acetylcholine is a neurotransmitter synthesized from the essential nutrient choline. Choline acetyltransferase is an enzyme that catalyzes the key step in acetylcholine synthesis, transferring an acetyl group from acetyl coenzyme A to the hydroxyl group of choline. Acetylcholine is used in both the central and peripheral nervous systems, but it is the most common neurotransmitter in the peripheral nervous system. All somatic motor neurons release acetylcholine to stimulate skeletal muscle contraction. Many of the neurons of the autonomic nervous system also release acetylcholine. For example, acetylcholine released from an autonomic neuron can regulate smooth muscle contraction. Nicotinic receptors are ionotropic receptors activated by acetylcholine. Although nicotinic receptors are nonspecific channels that are permeable to potassium, the concentration gradient and electromotive force drive sodium into the postsynaptic cell, leading to rapid depolarization of the postsynaptic cell following activation of a nicotinic acetylcholine receptor. Muscarinic receptors are metabotropic receptors activated by acetylcholine that produce varied effects depending on the receptor subtype. In the example shown here, muscarinic acetylcholine receptors stimulate contraction of pupillary constrictor smooth muscles in the iris. The catecholamine neurotransmitters are derived from the amino acid tyrosine. Dopamine, norepinephrine, and epinephrine are important examples produced by a series of enzymes. Aromatic amino acid hydroxylase adds a hydroxyl group to tyrosine-producing L-DOPA. Amino acid decarboxylase removes the carboxyl group from L-DOPA, producing dopamine. Dopamine beta hydroxylase adds a hydroxyl group to dopamine-producing norepinephrine. Phenylethanolamine N-methyltransferase adds a methyl group to norepinephrine, producing epinephrine. Norepinephrine and epinephrine are also known as noradrenaline and adrenaline. These synonyms are related to the fact that these chemicals are produced by the adrenal glands. When released into a synaptic cleft by a neuron, norepinephrine and epinephrine can function as neurotransmitters, but the same chemicals can also function as hormones when they are released into the blood by the adrenal glands. Norepinephrine is an important neurotransmitter in both the central and peripheral divisions of the nervous system. This is a key neurotransmitter used by motor neurons in the sympathetic division of the autonomic nervous system. Norepinephrine binds adrenergic receptors, all of which are metabotropic, with alpha and beta subtypes linked to different second messenger signaling pathways. In this example, we see how norepinephrine activates alpha-adrenergic receptors to stimulate contraction of pupillary dilator smooth muscle cells in the iris. Glutamate is the main excitatory neurotransmitter that mediates communication between neurons in the central nervous system. Glutamate is an amino acid released from the digestion of proteins, so this chemical is abundant in all cells. However, large amounts of glutamate are needed for neurotransmission in the brain, so other amino acids can also be converted to glutamate. Glutaminase is an enzyme that catalyzes the synthesis of glutamate from the amino acid glutamine. As a neurotransmitter, glutamate stimulates both ionotropic and metabotropic receptors on postsynaptic neurons. AMPA receptors are ionotropic receptors that allow sodium ions to rush into postsynaptic neurons, producing excitatory postsynaptic potentials. NMDA receptors are calcium permeable ionotropic receptors that require both glutamate binding and depolarization before calcium ions can enter. Activation of NMDA receptors is important for a process called long-term potentiation that changes the strength of synapses during the process of learning to form memories. Repetitive stimulation of a neuron by multiple excitatory synapses will activate NMDA receptors. The calcium that enters neurons through NMDA receptors stimulates a slow response leading to increased synaptic strength. Increasing the number of AMPA receptors at the synapse is one mechanism that can increase synaptic strength for long-term potentiation. Gamma-aminobutyric acid, commonly pronounced GABA, is the main inhibitory neurotransmitter that works in opposition to glutamate in the brain. GABA is synthesized from glutamate in a reaction catalyzed by an enzyme called glutamate decarboxylase. Ionotropic GABA receptors open chloride channels to rapidly produce inhibitory postsynaptic potentials. There are also metabotropic GABA receptors that indirectly open potassium channels for slower, longer-lasting inhibition. We can think of these two neurotransmitters as being analogous to traffic signals. Glutamate is like the green light of the brain that stimulates neurons to speed up signaling by producing excitatory postsynaptic potentials that lead to action potentials. GABA is like the red light of the brain that slows down neural communication by producing inhibitory postsynaptic potentials.